Well, good morning. Today we're covering the physiology of the heart. It is a hybrid between two other videos that are on my YouTube channel. The two videos can be found under Physiology Lecture Unit 3 if you want a more detailed but longer lasting lecture. One is called the cardiac cycle, and the other one is called contractile and specialized cells, or specialized cells and contractile cells. The specialized and contractile cells is an hour and 9 minutes, and the cardiac cycle one is 57 minutes. So it's a hybrid of what I'm doing for you today between those two lectures. And even though I have tried to whittle those two lectures down, it still is going to be a pretty intense lecture. We have a lot to go through. We have to talk about the cardiac cycle. We have to talk about the beast, which I'm sure you've seen in your homework packet. I'll explain what that is. And we have to talk about different types of action potentials. Now, the disadvantage for me doing this semester to you all is you had neurophysiology and anatomy last semester, didn't you? So it's really a shame that they don't have A&P1 and A&P2 where you are dealing with excitable cells all in the same semester, because it's the same thing. I really need you to go back and remember, because I know your instructor, and I know she taught you this, the difference between voltage-gated channels and ligand-gated channels. I know she did. I can't speak for Ray, but I know for the most part your instructor did. Thankfully, you just had a test, a lab test on the heart, so you know all the regions of the heart. You know the different chambers. Now we have to appreciate the cellular anatomy of cardiac muscle, but also the physiology of it. Good morning. Cardiac muscle is striated. It is mononucleated for the most part. Some textbooks will say mono to dinucleated, but for testing purposes, we're going to say mononucleated, okay? It is striated, which means that the actin and myosin filaments, myofilaments, are actually arranged in a parallel fashion, just like you learned for skeletal muscle physiology. They are bigger than smooth muscle, and they also have these structures called intercalated discs. And this allows the action potential, again, that's last semester, the electrical event always has to precede the mechanical. Yes, you've heard me say that so many times from last semester, bless you. The intercalated discs allow the action potential, the electrical event, to spread from one cardiocyte, that's the general term for these cells, to the next one. And because they are pretty large fibers, they don't really have a lot of resistance to the spread of this electrical event. And that means that the heart can beat in syncytium. That's a hard word, but that means together. But it's not all four chambers contracting at once. That can't happen. We don't want that to happen. You see, blood will only move from high pressure to low. I taught you that also when we were discussing vessel anatomy and physiology. I went through the whole thing many times. If you want blood to go out of the ventricles have to generate more pressure than the blood that's already in the elastic arteries just outside the heart, like the aorta and pulmonary trunk. You want blood to flow from the aorta, if we just look at the left side, to muscular arteries. The muscular arteries have to have a lower pressure than the elastic arteries. We went through that. Same thing with blood going to, through, and out of the heart. Valves will open and close, as you will see, due to pressure changes, not because of muscle contraction. They will open and close due to pressure changes. That is fundamentally why the blood will flow. The valves opening and closing ensure one-way movement of blood to, through, and out of the heart. That's their role. But the reason why the blood flows is because of pressure. And that goes to Ohm's law. That goes back to Ohm's law back in last semester, and we can apply it to flow. The equation for flow, and I showed you this on our blood vessel, is Q equals change in pressure over resistance. Nowhere in this equation do you see valves opening or closing. You don't see that. The valves opening and closing, they open and close because of pressure changes. That's it, period, end of story. And blood, their job is to ensure one-way movement. There are two different types of cardiocytes. Again, both of them can contract. One is called a specialized cell and one is called a contractile cell. They also have different names in other textbooks. The specialized cell is also referred to as the leader cell, and the contractile cell is sometimes referred to as the follower cell. The reason why leader and follower might be better terms to use is because when I use specialized and contractile, the student thinks that it's only the contractile cells that contract. That is not true. Both types of cells do contract. They both do. The contractile cell is called that because it is the majority of the myocardium, and I don't need to remind you what that is, right? No. It is the majority of the myocardium, so they do the heavy pumping. The specialized cells are special because they can spike an action potential on their own. They are not nerves. They are not nerves. But they can reach threshold on their own. They can fire that bullet, as I taught you last semester, on their own. And I have to tell you today the molecular reason why. Without the specialized cells, the contractile cells would just sit there going, nothing to do here. They can't spike an action potential on their own. And it's because of the intercalated discs that when the specialized cells do spike the action potential, that electrical event spreads from fiber to fiber, from chamber to chamber. And this allows the atria, then the ventricles. Atria, ventricles. Atria, ventricles. When the right atrium contracts, the left atrium contracts. They contract before the ventricles. When the right ventricle contracts, the left contracts. Atria, ventricles. So we need to appreciate the anatomy and the physiology. Why do we get this delay between the atria, and the ventricles? Why do they get their time first and then the ventricles? Timing is everything. Timing is everything. I can't just vomit on you and explain, so bear with me. Here we go. I'm going to go through all of this. There is a fibrous insulator. Dare I say, some textbooks refer to it as the skeleton of the heart. Now, your heart does not have bones in it, of course not, but there is a fibrous skeleton. And I remember watching this science program with my son, both my sons, and in this program, hold on, let me. In this program, I was on Zoom. I'm like, I wonder how many of my other students were watching this right now. In this program, they actually used an enzyme to digest away all of the cardiocytes, leaving behind only the fibrous insulator. So it was beautiful. It was like this lattice of fibrous collagenous proteins, and you can see the scaffold that it created. And then they were able to actually repopulate the muscle cells, the cardiocytes, onto that scaffold, which opens the door to heart transplants where you don't necessarily have to wait for a donor. You could, in theory, take the person who needs a new heart, grow their own cardiocytes on the scaffold, and then here we don't have to worry about being compatible with their immune system. That's amazing. This fibrous insulator, though, just so you know, does not conduct electricity very well. It doesn't. It gives us some resistance. And it's just enough, along with a few other factors that I will explain, where it allows the atria to have their electrical event and then their mechanical event, contraction. And then the ventricles get their turn. They get their electrical event, and then they get their mechanical event. This ensures that blood flows to, through, and out of the heart in a one-way direction. Let's just take a moment to watch that. I just said a whole bunch of things. Just take a moment to watch this video a few times. And then I'm going to describe what you should be seeing, and then I want you to take a moment to watch it again. First, in yellow, are specialized cells. They start the excitable event, the electrical event. Watch the action potential spread from one yellow group to the next. Electrical events precede mechanical. Notice that the atria get their electrical event first, and then they contract. Finally, the electrical event reaches the ventricles, and then they get their turn at the mechanical event. Can you see that? So I'm going to tell you about the specialized cells first. Everything you see here in yellow is a specialized or leader cell. Anything that you're seeing in the light gray color is a contractile or follower cell. Most of the myocardium anywhere in the heart is the contractile cells or follower cells.
The gray are what now?
The light gray would be the contractile, also known as follower cells. I'm going to tell you about these delays. If you look carefully again, you'll see a spot where it seems like the electrical event kind of pauses and then moves again. Can you see it? Right there? Okay. That is one place where we have a delay. Another, go ahead. No, ask.
Is that why it's to let the other atrium through?
Well, they both contract at the same time.
Oh, okay.
And I'm going to talk about, see this over here? See, that pathway is called an internodal pathway. I'm going to address all of this one slide at a time. But there's also, do you see this right here? That group of specialized cells is called the AV bundle, also known as the bundle of His. There is also a delay there. It's going through that fibrous skeleton that I told you about. You all were tested on the interventricular septum, right? That was in your hit list. Well, are you okay? Did you almost shoot coffee out your nose?
I was driving home on Monday and I'm like, interventricular septum.
It came to you. A little too late. But there's also an interatrial septum, and there's also an atrial ventricular septum. You see, you can almost draw the heart in my poorly drawn way so that you can see the 4 chambers. And wherever you have that cross pattern, that's a septum. That means wall. That means fibrous skeleton. And the electrical vent has to get through that. So we're going to see two places where there is a delay. And they are small, short-lived delays, but it's enough. Bless you. The atria get their electrical event, then mechanical blood flows from the atria to the ventricles. Then the electrical event gets to the ventricles, and they have their mechanical event, and the blood flows from the ventricles out to either the pulmonary trunk, which comes off the right ventricle, right? Correct? Or the aorta, off the left ventricle. So specialized cells, again, can reach threshold on their own. That means they can spike an action potential all on their own. They are not nerves. Well, Kara, we learned last semester about the sympathetic and parasympathetic division and how sympathetic can accelerate heart rate and parasympathetic can decrease heart rate, and that's true. Those are nerves. They will influence these specialized cells. I'm going to remind you of that in just a few slides. The nerves can influence how rapidly the specialized cells can reach threshold or decrease how quickly they get to threshold. Contractile cells would be at a resting membrane potential their entire lifespan if it weren't for those connections between the specialized cells and the contractile cells. In one of those videos, the one that's an hour and 9 minutes, I described to you how I actually took fetal mice hearts, minced them up, and plated the cardiocytes very sparsely on a petri dish. And because I took them so early, they were still mitotically active. And it was fun for me to watch them when they were first plated down. Some of the cells would ball up and then flatten out. And other cells, they were still alive. You can tell that because they kind of glow under a light microscope. They were healthy, they were living, but they didn't ball up at all. And over the course of a week or so, the whole plate was confluent. That's what we say. It was all touching. And then the whole sheet of tissue would ripple like this. And it was because those cardiocytes made connections, the intercalated discs, and now every cell was pretty much connected to its partners, even ones on the other side of the dish. And the whole thing rippled in syncytium, togetherness. Syncytium. S-Y-N-C-Y-T-I-U-M. Syncytium. I thought I had that. There you go.
There it is. And triggers. These words are incredible. I know.
Well, I mean, if you play Wordle, you never know. I mean, even though syncytium is more than 5 letters and 6 letters, but still, it helps. So let's go through the specialized cells first. The sinoatrial node, referred to as the SA node, and sometimes referred to as the pacemaker, is up here in the upper right atrium. It's called the pacemaker because the cells there will reach threshold the fastest. And from there, it ripples through the atria, down through to the ventricles. It sets the stage. I am the pacemaker of this class.
Mm.
I set the exams. I say, we have to do this. Let's go, let's go, let's go. Correct? If you were the pacemaker, you'd still be way back on learning about white blood cells. That can't happen. We have to move forward.
Sure.
Yes.
Is that what they replace?
Yes.
Sure.
Yes. So it can fail. We're going to talk about that. And we're going to talk about what their action potentials look like. So I'm going to try and get you to remember what a action potential looked like in a neuron. And I'm going to remind you of what it looks like in the skeletal muscle cell. We actually need to be mindful of their different profiles, what they look like, to appreciate that the heart, under normal physiologic conditions, does not reach tetany. Skeletal muscle can. You did a Physio X on that. You did as extra credit. Maybe you need to revisit that. It was Physio X number 2, I want to say. I'll look that up. Hold on. Yep, Physio X 2. So don't worry, we're going to review all of that. From the SA node, the action potential, the electrical event, spreads down to the left, but also down to the AV node, atrioventricular node. Its name tells you it's between the atria and the ventricles. We are going to see one of our 2 delays here. The cells of the AV node are small, that means more resistance, and they have fewer intercalated discs. That is a good thing because it allows some delay for the atria to have their events before the ventricles have theirs, because blood only flows from where to where? High pressure to low pressure. Okay, so I can't film this. I'm just going to pause it.